Hello everyone and welcome back to the Clinical Cousins YouTube channel. Today we are going to go over the excitation and contraction coupling of our smooth muscle cells. An important difference between smooth muscle cells and skeletal muscle cells is that smooth muscle cells, or SMCs, do not need a motor neuron to initiate their action potential. Also, SMCs do not have troponin. This is clinically important because one of the ways that we assess if there has been damage done to the heart during a heart attack is if there are elevated levels of troponin from cardiac or skeletal muscle present in our blood. Our SMCs use instead a calcium-binding protein that is conveniently named calmodulin. Now, the sequence of excitation-contraction coupling in smooth muscle cell is as follows. Calcium from the extracellular fluid comes in through our SMCs through these calcium leaky channels. Interestingly, our SMC can use this tiny bit of positive potential and not a whole action potential's worth to open up its voltage-gated calcium channels. When this occurs, calcium will rush into our SMCs and cue our ryanodine receptors to open the sarcoplasmic reticulum. Remember that our sarcoplasmic reticulum is packed with calcium, and when it is open, our SMC will receive this intracellular calcium shower. This calcium will shower our calmodulin. When calmodulin binds to calcium, it does two things. First, it removes proteins called calponin and caldesmin, which are covering our globular actin. Second, it activates an enzyme called myosin light chain kinase, or MLCK. This enzyme is a kinase connector that connects or phosphorylates and activates the light chain proteins on the thick myosin heads. Once these light chains are phosphorylated or turned on, our myosin will use ATP energy to form cross bridges with each of our globular actin. This whole process will come to a halt when the intracellular calcium is accumulated by our humble calcium ATPase cleaner upper. This calcium ATPase cleaner upper is constantly throwing the calcium from the inside of the cell back into the SR calcium sac. As always, thank you for taking the time to learn with us today, and remember to like and subscribe for more content.